Hello, welcome to Care ALS Plus Capsule Episode 4.
Yes, welcome.
And today we are doing a deep dive into Parkinson's disease.
Yes.
It is a very complex neurodegenerative disorder,
Yeah.
but we want to try and make this clear and understandable for everyone.
Absolutely. And it's actually the second most common neurodegenerative condition,
Wow.
affecting about 2 to 3% of people over the age of 65.
I see.
So it's a huge area of research, and we're going to try to give you a really solid overview of what we currently know.
Yeah, we really want to take this deep dive and go through all of the fundamentals of Parkinson's. So we're going to talk about what causes it, how it develops in the body, which we call pathophysiology.
Right.
The markers that might help us diagnose it, which are known as biomarkers,
Yeah.
and the treatments that are currently available.
Exactly.
So really a 360-degree view of this.
Yep.
And we're going to try to keep this straightforward
Absolutely.
and easy to follow.
Yeah. Great.
Okay. So let's start off by understanding really the roots of Parkinson's and how it unfolds.
Mhm.
I mean, it's actually really remarkable to think that James Parkinson first described this condition
Yeah.
around 200 years ago.
Yeah.
And that was really initially just focused on the motor symptoms.
Right.
And we've come to realize since then that it is much more than just a movement disorder, right?
Right, absolutely. It's a very complex multi-system disease.
Yeah.
So yeah, we've come a long way from 200 years ago when it was just really considered a movement disorder. It's really much more than that. And I think that's a really important point to make, is that it affects so many different systems in the body.
Yeah.
And so that's why it's so complex, and there's still so much that we're learning about it.
Yeah, because when you think of Parkinson's, right, you often think of the tremors. That's sort of what we think of.
Right.
But there's so much more to it, including things that aren't directly affecting movement.
Right.
And the research has really shown, we've been talking about some sources and some research articles, they have really highlighted that there's a wider range of issues involved, right? Things like changes in thinking, sleep disturbances,
Yeah.
and even a reduced sense of smell, which is very interesting.
Yeah, that's right. All those non-motor symptoms, are really prevalent, and they have a huge impact on quality of life for people with Parkinson's.
Absolutely.
So I think it's important to address those as well. When we think about the kind of core pathophysiology of Parkinson's, what's happening in the brain, there are really two key features. One is that you get a loss of nerve cells in a region called the substantia nigra. You can think of this as a key area that produces dopamine. And that results in a shortage of dopamine in another area of the brain called the striatum, which is really critical for movement. And the second key feature is that we get these abnormal clumps of a protein called alpha-synuclein within these nerve cells, these what we call Lewy bodies and Lewy neurites.
Okay. So it's really this combination of the loss of these dopamine-producing cells,
Right.
but then also the accumulation of this alpha-synuclein, right?
Right.
That really defines what we would call idiopathic Parkinson's disease.
Exactly. Yeah. So you need both of those features for a definitive diagnosis of idiopathic Parkinson's disease. And it's interesting, this loss of dopamine neurons in the substantia nigra, it doesn't happen uniformly. It actually tends to start in a particular part of the substantia nigra, and then it kind of gradually spreads.
Interesting.
And what's really, I think, fascinating is that this damage likely begins years before the motor symptoms even start to appear. So there's this long kind of pre-symptomatic or silent phase of the disease where this damage is happening, but people may not even realize it yet.
That's such a crucial point.
Mm.
And while most Parkinson's cases seem to occur randomly, the sources tell us that about 5 to 10% actually have a genetic link.
Yeah, that's right.
And so how does this hereditary aspect, how does that help us understand the disease in general?
Well, it's been incredibly valuable to study these hereditary forms of Parkinson's, because by identifying the genes that are involved, we've actually been able to gain insights into the critical molecular pathways that are disrupted in all forms of the disease,
Interesting.
whether it's inherited or not.
Okay.
So some of these key pathways that are involved in Parkinson's, both sporadic and hereditary, have to do with how the body manages this protein, alpha-synuclein,
Yep.
the health and function of mitochondria, which are the energy powerhouses within cells, protection against cellular damage, which we call oxidative stress, maintaining the right balance of calcium,
Right.
transporting essential materials within nerve cells, and then the role of inflammation in the brain.
Okay, so we're gonna unpack all of that.
Yes.
So let's zoom in a little bit on this alpha-synuclein because we keep talking about it.
Right.
Um, we know that it clumps up in Parkinson's,
Right.
but what is its normal job? Like, what's it supposed to be doing in the brain?
Yeah, so alpha-synuclein is really a central protein in this whole story. So mutations in the gene that makes alpha-synuclein, we call SNCA, can actually directly cause hereditary Parkinson's.
Oh, wow.
Um, and even in sporadic cases, genetic studies have linked, variations near this gene to an increased risk of Parkinson's and also to higher levels of alpha-synuclein.
Okay.
So normally alpha-synuclein is found at the connections between nerve cells, and it's thought to play a role in how these connections function.
Yeah.
We don't know exactly what its role is, but it's definitely involved in that process.
So it's supposed to be helpful. It's a helpful protein.
Right, exactly.
And then in Parkinson's, it just starts misbehaving.
Right, exactly.
So what exactly happens?
Yeah, so in Parkinson's disease, this protein alpha-synuclein, it changes its shape. We say it misfolds, and then it starts to stick together. And so it forms these small aggregates called oligomers.
Okay.
And then those can come together to form these longer strands called protofibrils.
Okay.
And then eventually you get these larger insoluble fibers that make up these Lewy bodies and Lewy neurites that we talked about earlier.
Okay. So it starts clumping up.
Right.
Now, is there something that's supposed to be clearing these out, these misfolded proteins?
Yeah, so our cells actually have these built-in kind of recycling and waste disposal systems.
Right.
And the two main ones are called the lysosomal autophagy system, or LAS, and the ubiquitin proteasome system.
Okay.
And in Parkinson's, these systems don't seem to be working as efficiently. And that could be due to a number of things, maybe due to the aging process itself.
Yeah, that makes sense.
But that can lead to this buildup of alpha-synuclein. And it becomes this kind of vicious cycle because the accumulated alpha-synuclein can further interfere with these very clearance systems that are supposed to be getting rid of it.
Right. Like it just builds up and up.
Exactly.
Now, some sources have mentioned some specific genes that are linked to this problem with this protein clearance.
Right.
And there are a few that they mention, like LRRK2, GBA, and VPS35.
Right.
Can you explain, what's the connection there?
Yeah, so these are, some of the genes that have been implicated in Parkinson's. So, for example, a common mutation in the LRRK2 gene, we call it the G2019S mutation, impairs the LAS.
Okay.
And that contributes to the accumulation of alpha-synuclein.
Okay.
And mutations in the GBA gene are actually the most common genetic risk factor for Parkinson's.
Oh.
So GBA codes for an enzyme in lysosomes, which are a key part of this LAS, this lysosomal autophagy system.
Right.
And when GBA is not working correctly, it can lead to the buildup of various substances, including alpha-synuclein.
Okay.
And similarly, mutations in the VPS35 gene, which is involved in transporting proteins within the cell, can also disrupt the proper handling of alpha-synuclein
Okay.
and impact lysosomal function. And what's really interesting is that, issues with another protein called ATP13A2, which is also involved in lysosomal function, can even influence the effects of these LRRK2 and GBA mutations.
Oh, wow.
So it suggests that, all of these proteins are really kind of part of this interconnected cellular machinery.
Right. They're all working together.
Right. Exactly.
Now, we also have this idea of, it's kind of a big term, but it's called prion-like propagation of alpha-synuclein.
Right.
And that's a strange idea. What exactly does that mean for Parkinson's?
Yeah, so it's a hypothesis that suggests that once these alpha-synuclein aggregates form in one nerve cell,
Okay.
they can actually somehow spread to neighboring cells.
Interesting.
So you can kind of think of it like a seed.
Right.
That can kind of cause more misfolding and aggregation in new areas.
Right.
And so the way that this might happen is that these aggregates can travel along the long projections of nerve cells. They can be released outside the cell and then be taken up by other neurons where they can then trigger more alpha-synuclein to misfold and clump.
Okay.
And if the LAS is not working well, it may even increase the release of this problematic alpha-synuclein.
Oh, okay.
So that's kind of how we think this might be happening.
So it's really like a domino effect.
Yeah.
One cell affects another cell.
Exactly.
Okay. And how does this connect to things like, problems with smell
Right.
or constipation,
Right.
which are, some early symptoms of Parkinson's?
Yeah, so this could explain how this alpha-synuclein pathology seems to spread through different parts of the brain over time.
Okay.
And it potentially starts in areas like the gut or the olfactory bulb, which is involved in our sense of smell.
Okay.
And that might be why we see some of these early symptoms like loss of smell and constipation appear before the more obvious motor issues.
Okay, that makes a lot of sense. Now let's move on to mitochondrial dysfunction. So we know that mitochondria are essential for energy production in cells,
Right.
but what happens to them in Parkinson's?
Yeah, so there's really substantial evidence linking problems with mitochondria to Parkinson's.
Okay.
And so we often see reduced activity of a key mitochondrial component called complex 1.
Okay.
And additionally, genes controlled by PGC1 alpha, which is a master regulator of mitochondrial health, tend to be less active in PD. And, while small amounts of alpha-synuclein might normally be present in mitochondria, it seems that excessive buildup can directly damage complex 1 and lead to increased cellular stress.
Oh, yeah.
So it appears to be another kind of self-perpetuating cycle where alpha-synuclein problems and mitochondrial problems worsen each other.
Right.
Um, and interestingly, even in animal models where researchers use toxins to damage mitochondria,
All right.
they can observe features that are similar to Parkinson's.
Okay.
And genetic models, for example, there's this mouse model called the mitopark mouse.
Okay.
And these mice have specific mitochondrial impairment in dopamine neurons.
Okay.
And they also develop Parkinson's like changes in the brain.
Well, yeah.
And that really points to the importance of mitochondrial function in this disease.
Yeah, yeah. And so this mitochondrial dysfunction, could that play a role in the early damage we see to the long nerve fibers in the brain?
Yeah, that's right. So the lack of sufficient energy or the physical blockage caused by these alpha-synuclein aggregates hindering transport along these axons
Hmm.
could lead to early problems in these connections between the substantia nigra and the striatum.
Okay.
And some studies have actually shown that, you can detect changes in the striatum even before a formal Parkinson's diagnosis is made.
Oh, wow.
And some of these genes that we've talked about, like LRRK2, PARK2, and PNK1, which are, known to be involved in Parkinson's,
Right.
also play really crucial roles in mitochondrial function and the process of mitophagy, which is the way that cells clear out damaged mitochondria.
Okay.
And even the breakdown of a protein called MRO, which helps to link mitochondria to the cell's transport system,
Okay.
seems to be affected in Parkinson's.
Okay.
And that can further impact mitochondrial health and potentially lead to energy deficits.
Yeah. So many different factors are involved in this.
Right.
Okay, so now let's talk about oxidative stress. It's a term that we hear often
Yeah.
in the context of brain diseases, but what role did it play in Parkinson's?
So we find clear signs of increased oxidative stress in the brains of people with Parkinson's.
Okay.
And this is likely a consequence of the mitochondrial problems that we just discussed.
Right.
And mutations in the DJ1 gene, which is thought to have protective antioxidant properties and is linked to early onset Parkinson's, are also associated with increased oxidative stress in cells.
Oh.
So it seems that the dopamine-producing neurons in the substantia nigra seem particularly vulnerable to oxidative stress for a few reasons.
Okay.
They have very long axons that require a lot of energy.
Right.
They have this natural kind of rhythmic firing pattern that involves energy-consuming calcium cycling.
Okay.
And then dopamine itself, as well as its breakdown products, can actually be toxic.
Oh, interesting.
And importantly, oxidative stress can also further impair the protein clearance systems that we talked about earlier. So it really highlights how interconnected all these pathways are.
Right. It all goes back to that.
Right, exactly.
So it's all related.
It's all related, yep.
Okay. And then what about neuroinflammation? So we hear about inflammation in so many conditions.
Yeah.
What's its role in Parkinson's?
Yeah, so neuroinflammation is definitely a key feature in Parkinson's.
Okay.
We see evidence of this in post-mortem brain tissue and
Yep.
brain imaging studies,
Okay.
and then also an analysis of fluid biomarkers.
Okay.
While it might not be the initial trigger of the disease, it likely plays a significant role in its progression.
Okay.
And we've observed that these dopamine-producing neurons in Parkinson's brains can show increased levels of certain proteins, and this can make them targets for immune cells.
Oh, wow.
Genetic studies have also linked Parkinson's risk genes, such as LRRK2, to immune function.
Okay.
So it appears that this buildup of alpha-synuclein can actually activate both the innate and adaptive immune systems in the brain. And then the resulting inflammation may then further promote alpha-synuclein misfolding, so it creates another vicious cycle.
Okay, okay.
And even the bacteria in our gut are being investigated for their potential influence on this brain inflammation and alpha-synuclein pathology.
Wow.
But, it's important to remember that the immune cells in the brain, like microglia, can
Right.
also try to clear out alpha-synuclein.
Right.
So the role of inflammation is complex. It likely has both harmful and potentially protective aspects.
So it's kind of a double-edged sword.
Yeah.
You know, it could be good or bad.
Right.
Okay, so all of these factors that we've been talking about, alpha-synuclein,
Yeah.
mitochondrial issues, oxidative stress, inflammation,
Right.
they're all contributing to the loss of these dopamine neurons.
Right.
And then ultimately, the motor symptoms that we associate with Parkinson's.
Right. Exactly.
So can you explain a little bit about what's actually happening in the brain circuits that control movement? Like what's happening there?
Yeah, so the basal ganglia, it's this group of brain structures that's crucial for regulating movement
Okay.
and coordinating goal-directed actions.
Okay.
And it does this through its connections with the brain's outer layer, which we call the cortex,
Right.
and a relay station called the thalamus.
Okay.
In Parkinson's, this reduced dopamine in the striatum actually disrupts this delicate balance within these basal ganglia circuits.
Okay.
And so this leads to an overactivity of one pathway, which we call the indirect pathway, and then underactivity of another pathway, which we call the direct pathway.
Okay.
And ultimately, this results in an increased inhibition of what we call the thalamocortical projections, which are essential for initiating movement.
Oh, I see. Okay.
So there's also a more direct pathway called the hyperdirect pathway that may play a role in stopping movements.
Yeah.
And then we also see changes in the kind of rhythmic electrical activity of the brain,
Right.
and particularly an increase in a specific type of activity called beta band activity.
Okay.
And this is often seen when someone with Parkinson's is in an off state,
Okay.
meaning their medication isn't working optimally.
Right.
And this abnormal activity tends to decrease when dopamine medication or deep brain stimulation is effective.
Okay.
So it suggests that beta band activity might be a marker of this kind of dopamine depletion state.
Right. Okay. And then, beyond these movement-related issues, the non-motor symptoms are a really significant part of the Parkinson's experience.
Absolutely.
Yeah.
Yeah. And they become increasingly common as the disease progresses.
Right.
So things like, difficulties with sleep, changes in thinking and memory,
Right.
mood disturbances like depression and anxiety,
Right.
problems with the autonomic nervous system, which controls things like blood pressure and digestion.
Right.
All of these can really significantly impact a person's overall quality of life.
Right. Absolutely.
So it's really important to address those as well.
Yeah, it's not just about movement. It's really it's a whole body, disorder.
Yeah, exactly.
Okay. So this is giving us a picture of a really intricate disease.
Yeah,
So with all this understanding of the underlying mechanisms,
right.
where do we stand with actually diagnosing Parkinson's?
Yeah.
Are there reliable tests available?
That's a great question. And I think it's important to acknowledge that, currently the diagnosis of Parkinson's disease is still primarily based on a clinical evaluation.
Okay.
So that means a neurologist assessing the presence of these characteristic motor symptoms that we've talked about.
Right.
But, this clinical diagnosis can be challenging, especially in the early stages of the disease.
Right.
And even in specialized centers, there can be a degree of diagnostic error. So this really underscores the need for more objective diagnostic tests and biomarkers.
Yeah, that makes sense. So what kind of brain imaging techniques are being used or explored
Yeah.
to help with this diagnosis?
Yeah, so there are several neuroimaging techniques that play a role.
Okay.
So one is PT scans,
Okay.
and they can visualize how well dopamine is being taken up and stored in the striatum, which is often reduced in Parkinson's.
Okay.
Another type of scan is called a SPEC scan.
Okay.
And these use a tracer called 123I ioflupine. It's often referred to as a DAT scan,
Okay.
and it can help to distinguish Parkinson's from other conditions that might look similar but don't involve the same loss of dopamine function.
Okay.
And then standard structural MRI scans are important to rule out other potential causes of someone's symptoms.
Right.
More advanced MRI techniques are also being investigated to see if they can detect subtle changes in the basal ganglia in people with Parkinson's. MIPI SPECT is another useful tool that can assess the health of the sympathetic nervous system in the heart, which is often depicted in Parkinson's, but not in many of its mimics.
Okay.
And finally, there's promising research using a specialized type of MRI called neuromelanin imaging.
Okay.
And this can help to visualize the substantia nigra more clearly
Okay.
and may aid in early and more accurate diagnosis.
Okay. Interesting. Now, what about what about genetics? We talked a lot about the hereditary forms.
Yeah.
But can genetic testing be helpful for diagnosis in general?
So we've now identified this growing list of genes that can directly cause Parkinson's.
Yeah.
And we also know of other genes like GBA that can increase the risk of developing the sporadic form of the disease.
Okay.
Large-scale genetic studies have also identified many common genetic variations that individually have a small impact on risk.
Okay.
But routine genetic testing isn't typically part of the standard diagnostic workup for Parkinson's right now,
Okay.
except in specific situations. So, for example, if there's a strong family history of Parkinson's,
Right.
or if the disease starts at a younger age.
Okay.
And currently, the results of genetic testing don't usually change the immediate treatment approach for most people with Parkinson's.
Okay.
But, this field is evolving rapidly.
Yeah, it feels like
And as we learn more about how different genetic variations affect the course of the disease, and as targeted therapies are developed for people with specific mutations,
right.
for example, there are LRRK2 inhibitors that are being developed for people who carry LRRK2 mutations.
Okay.
Genetic testing is likely to play a more significant role in the future.
Yeah, that makes sense.
Yeah.
So what about tests on cerebrospinal fluid or
Yeah.
blood?
Yeah.
Those would be less invasive than brain imaging.
Right.
So are those tests helpful?
So researchers have extensively studied various proteins in cerebrospinal fluid, CSF, and blood, including different forms of alpha-synuclein as potential biomarkers for Parkinson's. Unfortunately, so far, no single CSF or blood test has demonstrated sufficient accuracy and reliability to be used as a standalone diagnostic test in clinical practice.
Okay.
So while some blood markers, for example, lower levels of a protein called apolipoprotein A1
Okay.
have been observed in association with disease progression, they're not really reliable enough for diagnosis.
Okay. So it's really tough to diagnose Parkinson's early on.
Yeah.
So what about the possibility of screening or even preventing the disease?
Yeah.
Since these brain changes seem to start so early on,
Right.
are there ways to identify people that might be at higher risk?
Yeah, this is a very active area of research.
Okay.
And so scientists are looking at various factors and subtle changes that seem to be associated with higher likelihood of developing Parkinson's later in life.
Okay.
So these include very subtle motor changes that might not even be obvious to the person,
Okay.
a reduced sense of smell,
Right.
chronic constipation, and then a sleep disorder called REM sleep behavior disorder, or RBD.
Okay.
And the International Parkinson and Movement Disorder Society has even developed research criteria to define a prodromal phase of Parkinson's,
Okay.
which is before the full motor symptoms appear.
Right.
And it's based on a combination of these risk factors and early signs.
Okay.
Researchers are also investigating other potential biomarkers, such as the levels of uric acid in the blood, combining genetic risk scores with other clinical factors,
Right.
analyzing changes in the gut bacteria,
Yeah.
and using advanced omics techniques to study proteins, metabolites, and RNA in biological samples.
Okay.
Interestingly, there's also research exploring the possibility of detecting alpha-synuclein pathology in the peripheral nervous system through biopsies of the skin, salivary glands, and gastrointestinal tract as potential early indicators,
Right.
even in individuals with idiopathic RBD.
Okay.
And then finally, neurofilament light chain, or NFL, which is a marker of nerve cell damage, is being investigated in blood and CSF as a potential indicator of neurodegeneration.
So now let's switch gears and talk about therapeutics.
Okay.
So what are the current treatments for Parkinson's disease?
Yeah, so I think it's important to start by saying that while we have effective treatments to manage the symptoms of Parkinson's, we don't currently have a cure.
Right.
And the disease is progressive.
Right.
So the primary goal of current therapies is really to address this underlying dopamine deficiency in the brain.
Right. Right. And that's where the medication LDOPA comes in.
Exactly.
Yeah.
Yeah. So LDOPA is still considered the most effective medication for replacing that lost dopamine.
Okay.
However, Its long-term use can lead to motor complications,
Okay.
such as fluctuations in its effectiveness and the development of these involuntary movements called dyskinesias.
Right. Okay.
So we've taken that in-depth exploration and, distilled what we hope are the most valuable insights for you.
Yeah, we touched on, potential initial causes, the complex chain of events,
Mhm.
the disease's progression, the exciting work in identifying biomarkers, and of course the current, therapeutic landscape.
Absolutely.
Yeah.
So reflecting on all of the different angles of Parkinson's disease that were covered in that really extensive discussion, what's one area that sparks your curiosity the most, something you'd want to explore in more detail? It just highlights how multifaceted this condition really is.
Absolutely. Yeah.
All right, thanks for joining us for this.
See you in the next episode of Carry LS Plus Capsules. Bye.